Hello everyone, today we will be talking about fermenter. What is a fermenter? What are the different parts of a fermenter and the different types of a fermenter? This topic is under your paper Industrial Microbiology. Now what is a fermenter? A fermenter is a closed cylindrical vessel that within that vessel you put a medium to which different microorganisms are added. These microorganisms will carry out its biochemical activity and convert the raw material into the industrially useful product. Now, what is the importance of a fermenter? The importance of a fermenter is that these fermenters are created in such a way that even using a very small amount of media, we can yield high amount of product or higher value product by using the microorganism. The first discovery of fermenter was during 1914 and 1915 by scientist Chain Wiseman. Later on in the year 1930, the large-scale aerobic fermenter was developed, followed by the year 1934, Scott and Schmidt discovered their aeration system. It modified the aerobic fermenter with the aeration system. It introduced the water and the steam inlet into the system. In the year 1950, Hindustan Antibiotic Limited in Pune developed the first industrial fermenter for India, and it is named as pilot fermenter. Now, what is the need or what is the objective behind designing a fermenter? Why do we need a fermenter? Mainly, we need a fermenter to produce different microbial biomass, to produce different microbial metabolites, to produce industrially effective enzymes, and to produce different recombinant products. So, while designing a fermenter, we should keep in mind several properties. The first is we should use a material, basically steel, so that it is not corrosive. It can withstand the weather condition. It should give high productivity. It should withstand the steam that is released during the sterilization process. And there should be control parameters to control the pH, temperature. There should be inlet and outlet pipes for recovery of the product. And the material that is used in the fermenter should be cheap and it should yield a very good or satisfactory product. Next is the design of a fermenter. You can see here, this is the design of a fermenter. If you consider the design of a fermenter, the fermenter has, we can consider the design into two categories. One is a basic part, another is a controlling part. First of all, let us talk about the basic part, that is the top plate. You can see here, this is the top plate. What is the function of a top plate? It covers the fermenter and it is generally made up of stainless steel. Next is the inoculation pipe, through which we introduce the inoculum or the microorganism into the fermenter. Then the drive motor. It drives the impeller shaft. What is an impeller? Impeller is nothing but a steerer. It helps in the homogenization of the medium along with the microorganisms. The third is the impeller shaft or the impeller. See, it holds the aggregator. This is the impeller. This is the impeller. And see here, this is the impeller. It acts as an agitation device. That means it helps in the proper mixing of the medium and the microorganism in a homogenized condition. Then there is a steerer. This is the steerer, the main pipe to which the impeller is attached. That is known as the steerer. The next is the baffle. These are the baffles. This part is known as the baffle. What is a baffle? What is the function of a baffle? The baffle prevents, basically they prevent the counterflow of the medium, as well as it helps in the breakdown of the bubble, the air bubble that is formed during the bioconversion. It also improves the aeration efficacy of the fermenter. Next is the spurger. A spurger is the one that supplies the oxygen inside the culture medium through perforated tube. This is the spurger. You can see here sterile air is being introduced. So this is the spurger. It has a perforated, there are holes through which the oxygen or the sterile air is introduced into the fermenter. Then drain point. What is a drain point? To withdraw the medium, to withdraw the cells or the medium for the continuous fermentation. Okay, so this is the harvesting or the drain point. This is the sampling point. You can see here, this is the sampling point through which sample is being introduced. Then there are cooling jacket. As this process, during the fermentation process, huge amount of heat is being generated, so the entire tube is being fitted externally with a cold water blanket. Like there are pipelines of cooling jackets. Whenever the vessel is heated, continuously cold water is being introduced into the cooling pipes to balance the heat generated during the process. Next is the controlling element. What are the controlling element and why is it necessary? There are several parameters that we should keep in mind while considering or while operating a fermenter. That is temperature, pH, oxygen supply and pressure. So P100, this is that monitors the temperature. This is you can see here, this is the temperature probe. Then there is a pH probe, that will maintain or that will detect the pH of the medium continuously. There is a foam probe that senses the foam formation. If there is huge amount of foam, it hinders the process of fermentation. Then oxygen sensor, the level of oxygen is being monitored. Heating pads, this provides heat to the medium, that is the required temperature that is needed for the fermentation to take place. Cold finger, it passes the cold water inside a vessel to cool the content. Rotameter, it provides variable airflow into the culture vessel. And then there are air pumps that will pump the air. There are air pumps that will pump the air, and peristaltic pump that pumps the acid, base and anti-foam into the medium. See, this is the peristaltic pump. This is the peristaltic pump that will pump the acid, base, anti-foam depending upon the required pH. Suppose we require a pH of 8 for the fermentation, and after the biochemical activity the pH is dropping to 6 or 7. So we add the pH here through this so that the uniformity is maintained. So what are the basic functions of a fermenter? It should operate for a long term. It should be capable of operating aseptically. It should provide adequate aeration and agitation for the uniform mixing of the sample as well as the microorganism. It should consume less power. It should have controlling panels such as temperature, pH, oxygen level. It should facilitate the incoming or the inflow of the inoculum and the medium inside the vessel. It should not allow excess of evaporation loss, that means excess of water loss should not be there, and it should be easy to clean. It should be easy to handle. harvesting time should be low, and it should require easy maintenance, and it should be also very much cost effective. Next is, what are the different type of a fermenter? There is basically 5 major type of a fermenter, that is the stir tank fermenter, airlift fermenter, fluidized bed fermenter, packed bed fermenter, and the photo fermenter. First is the stir tank fermenter. It is the most common type of fermenter that is used. Till now, whatever we discussed about the fermenter, that is the stir tank fermenter. That is, it controls a motor draft shaft. You can see the figure is almost the same of the general figure. It has the impeller and have the 1/3 diameter of the vessel. This is the impeller. The height and the diameter should be 1:3. Air should pass from the single orifice. This is the air inlet. It should pass from the single orifice. Okay. Then this is the effluent, that is the for taking out the product. What is the function? The basic function is the homogenization, supplementation of the solid material, and aeration in the medium, heat exchange, as well as in stir tank fermenter, rotating stirrers are used. They are either found, the rotating stirrer, you can see here the stirrer is either found at the top of the vessel or at the bottom of the vessel. There can be two ways, either it is the baffle and the stirrer, either it is present in the top or it is present in the bottom of the vessel. Now, what is the advantage? These stir tank fermenters are very easy to operate. There is a good temperature control, construction is simple, operation cost is low, that means it is cost effective, easy to clean, and it does not cause any damage to the microbial cell. The major disadvantage is you cannot use it for immobilized cell, as well as you cannot use it for industrial enzyme production. Apart from that, it has a very low volumetric productivity. So the second type is the airlift fermenter. What is an airlift fermenter? You can see here the design is quite different. There is a single container inside which there is a hollow shaft or the hollow tube. This is known as the draft tube. It is not present in an aerobic fermenter or stir tank fermenter. Inside the vessel there is a hollow tube that is known as a draft tube. There is a gas flow inlet at the bottom, air inlet at the bottom, that allows the oxygen to enter. The gas flow inlet is perforated and it allows continuous distribution of air within this draft tube. This doesn't have any mechanical stirrer, and as the name suggests, the air lifts the medium upward. There is no mechanical stirrer. The pressure of the air lifts the medium upward and helps in the internal liquid circulation. In this fermentation, it occurs at a fixed rate of volume as well as a fixed rate of circulation. Now, what is the advantage here? It ensures adequate mixing, consumption of energy is less, it is favorable for aerobic culture, it's much more simpler than that of a stir tank fermenter. It can be used for both free and immobilized cell. Stir tank, the disadvantage was it cannot be used for immobilized cell, whereas in case of airlift fermenter, you can use it for the immobilized cell. It is widely used for the industrial production of SCP, that is single cell protein, and there is no excess of heat generation. The chief disadvantage is mechanical steering is absent here though. The third type is the fluidized bed reactor. You can see here the top portion of this bioreactor is expanded. Why it is so? Because this expanded top head reduces the velocity of the fluid, and the bottom portion you can see here is very narrow compared to that of the head. Solid, why this is designed in such a way? So that the solid is retained within the vessel and the liquid is flown out. Gas is introduced, sterile gas is introduced from the bottom, and there is a suitable gas liquid solid phase. This is the gas liquid. Why this is known as fluidized bed reactor? Bed means there are certain polymeric particles. You can see here small and about beads basically are used. PVC chips or beads are used, to which the microorganism attach and form biofilm, and then they carry out the fermentation process. Okay, so these beads should not be very light or very heavy. Very light, it will keep on floating. Very heavy, it will settle down at the bottom. So should be very careful while selecting what type of material we are using for the bead. Okay, so what is the advantage? The advantage is it mixes the content uniformly, maintains uniform temperature, and produces high volumetric product, and there is little or almost no clogging. Then packed bed fermenter. You can see here, this is the packed bed. There is a bed. You remember trickling filter in case of wastewater treatment. It is almost, you can see here, the structure is almost similar to that of trickling filter. This is the inlet point for the nutrient, but very simple construction. Packed bed fermenter. This is the inlet of the broth. Just like trickling filter, you can see here the broth trickles down. This is the PVC chip that will act as a bed. The microorganisms will form biofilm. Nutrient broth will trickle down through this, and these microorganisms will use this nutrient broth and catalyze the production of the industrial product. The solid matrix that is used, it is highly rigid, compressible, and it can be porous or non-porous. This is generally used for the immobilized biocatalysis. And once the product is formed, it is taken out from the bottom. Very simple advantages. The operation cost is low. It is very simple, easy to clean, widely used. You can see a wastewater engineering, that is, it is the basic model of a trickling filter that I said already. You can continuously operate it. What is the disadvantage? There is no temperature control. There is no pH control. You cannot see what is the product that is being produced, and there is all chances that there is chances of high particle clogging, because after a particular time period, what will happen? The biofilms will start to clog. Microorganisms will grow at a higher rate, and these chips will be covered by thick biofilms, and gradually that will result in the clogging. So this is one of the chief disadvantage of the packed bed fermenter. The last one is the photofermenter. What is the principle of a photofermenter? In case of a photofermenter, we use the light energy, that is basically the sunlight energy we use, or in certain times we use artificial light also. These fermenters are basically used for the production of beta-carotene and xanthin. You can see here, these are the tubes, the solar tubes that are used, different types, different designs of the tube. So the bioreactor is basically made up of glass or plastic. It contains a single container, and within that container, the tubes or the panel that are known as the solar light trappers or the receivers that are introduced. What the microorganism do? The microorganism will the solar trappers will trap the solar energy with the help of a centrifugal pump. Once the solar energy is trapped, once there is adequate amount of solar energy or the sunlight, then it is cooled, and the microorganisms, specific type of microorganisms such as microalgae and cyanobacteria, these are all blue-green algae. This requires light energy for the survival. So these microorganisms in presence of the solar light, it will grow in presence of the solar light, and the product will be formed during the night. Product will be formed during the night. So what is the advantage of this? It yields a very high product. It has a higher surface to volume ratio. There is better gas control transfer, uniform temperature gradient, as because there are several tubes, so gradiently the temperature is monitored. Very expensive method, because these solar trappers are very expensive, and there are very much technical difficulty during how we can sterilize this fermenter. So this is all about your fermenter. What is a fermenter? What is the basic design of a fermenter? What are the types of a fermenter? And what are its advantage and disadvantage? I hope this topic will be beneficial for you. Stay home, stay safe. Thank you.